Well, I really appreciate it. Okay, let me just okay that.
Yeah.
Yeah, so I sort of I think the abstract that I sent, the title I sent is specifically the brainstem work. And I promise I will talk about the brainstem work. I'll actually talk about all of the brainstem work because it's just so like it's very simple, so it's very easy to talk about. And as a result, I've given this a bit more of a broad scope. So I've titled it the integrated brain, which is what I'm planning to title my thesis, sort of inside scoop, because I couldn't think of a better title because I've done like a lot of different stuff, and so I had a really hard time finding like one title that would kind of encompass the I'm a little bit scatterbrained, so I keep doing like once I do one thing, I have to do some other thing that's like not very related. And Martin van Heuvel, his PhD thesis is called the connected brain. So I sort of thought I'm in good company if I call it the integrated brain. Yeah, I mean, subject to change. But in any case, as we know, as you know, I'm sure, the brain is a multi-scale organ. This is sort of a kind of boilerplate introduction. You've probably seen this similar introduction many times. But if I sort of illustrate literally a hierarchy of spatial scales, we might start really small, something like gene expression, and go to the proteins, something like receptors, all the way to the synapses that are connecting cells. Now we're talking about different cell types and the way they're distributed. We get brain structure, which in my case, usually some sort of tractogram coming from diffusion weighted MRI. And finally, brain function, which could be measured using fMRI bold. So these are supposed to be red blood cells or any kind of time series of neural activity. But really, when we say function, we mean processes like, I don't know, language or perception or movement or like doing all three at the same time. So my work, to kind of remind myself and you about the integrated brain, is trying to understand how all these different levels of description in the brain are being integrated together, how they fit together. And there's some facts of this kind of work. One is that no one person can be an expert in everything. So, kind of a nature of my work is I'm not an expert in any one of these spatial scales that I'm studying. I'm not an expert in receptor or chemo architecture. I'm not an expert in gene expression. I'm not an expert in brain structure or function. I'm sort of, I'm the person who knows the experts and tries to like put together the information that they are very much experts in. And as a result of this fact that no one person can be an expert in anything, we have this history of studying neuroscience, studying the brain, which is quite siloed. So it makes a lot of sense, right? It's so complicated that we sort of divide and conquer. So different people are studying different things, and as a result, there's so much data in each one of these kind of environments, these niches. And as a result, Brad Wojtek mentioned this as being data rich but theory poor. There's a lot of data at every single one of these points along the hierarchy of spatial scale, but because we're not necessarily making contact to one another, there aren't very many unifying theories about, well, how is the brain actually working then once we start to integrate all these different levels of information. So I'm not an expert in everything, like even arguably anything. And I'm definitely not going to give you any unifying theories, but that sort of just pits the problem of where this sort of integrated neuroscience work comes in. So my very first project, I was linking receptor distributions to structure and function. And the story behind this, I come from a network neuroscience lab, and so I was thinking a bit about the link between structure and function, so just structure-function relationships in general. And I sort of thought there's a very obvious mediator here, which is these receptor distributions. And as we know, receptors, these are these proteins that are sitting at the cell membrane. They respond to a neurotransmitter receptor that's going to change the protein configuration, which is going to let charged ions flow in or out of the cell, changing the ability for the cell to fire an action potential. And we have the same thing for mid-hapatropic receptors. It's going through a G protein instead. But in both cases, we have this situation where these receptors are directly modulating the the ability for this neuron to fire an action potential, so its dynamics. And then if we think, neurons that fire together wire together, then also probably its connectivity. So I thought, well, at the micro scale, I think it's pretty well established the role of these receptors. But at the macro scale, can we find a relationship between how these different neurotransmitter receptors are distributed and structural and functional connectivity? Now, I didn't have any data, and so what I did was I sort of, or Bratislav and I kind of cold called, by which I mean just send emails to people that we didn't honestly necessarily know. And we said, you've been collecting this data and you've been working on this data. Would you be willing to share a group averaged map of this receptor distribution, and would you be okay if we made it publicly available? And going into this, I thought that I would be met with a lot more hesitation than I would. I don't know. I thought people would be much more protective of this data they've been collecting, and I was really surprised at how happy people were, and like not even just happy, but like kind of didn't care. They're like, yeah, of course, go take it, do what you want with it, like. And of course, you can make it open and whatnot. And so that was sort of nice. And my little like lesson about this is that if there's something that you kind of want, then just ask for it, and it will probably work. And so we contacted all these different groups, and we ended up putting together this atlas of 19 different neurotransmitter receptors and transporters using PET imaging. across 9 different neurotransmitter systems and a whole bunch of healthy controls. So these are all group average maps from different people. It's not the same people, and it's not the same location. It's not the same necessarily scanner. I mean, it's all PET imaging, but from all over the world. So it's kind of a bit of a collation of all this different types of data. But once we had this, we were able for each brain region, you can see my mouse, right? Yeah, great. Okay, for each brain region, you can make a fingerprint of its receptor density. So you can get a sense of, is it higher in some receptors and lower in some other receptors? And then you can correlate these two fingerprints for every pair of brain regions, and you get this region by region correlation matrix, just telling you how similar are two brain regions in terms of the receptors that they express. And the logic here was, well, if two brain regions are expressing similar receptors, I think of these receptors as like ears. They're listening for similar signals. So if you get one signal that goes out through the brain, and you have two brain regions that have similar receptor fingerprints, they're probably going to respond similarly to that signal. And so I'm thinking respond similarly as in similar dynamics, but also probably more likely to be connected. So we take this receptor similarity matrix and compare it to structural connectivity from diffusion weighted MRI and functional connectivity, so just the correlation of fMRI bold time series. And then we can ask very basic questions like, if two brain regions are structurally connected, what is my receptor similarity? Do I have a greater receptor similarity with regions that are connected? Same thing for if two brain regions are functionally connected, or sorry, within similar functional modules. So doing functionally similar things, am I more likely to have similar receptor similarity, or sorry, similar receptor distributions? And in both cases, yes, which sort of made sense to me. I thought that was kind of, I would hope so. And then here what we're doing is for every brain region, I'm trying to predict its functional connectivity profile, so like a row right here, from its structural connectivity profile. Because you guys know your stuff, this is technically communicability is what I'm using, not the structural connectome, but in any case. And then I try to predict function from structure, so that's on the x-axis, and then I include receptor information, so I include a row of that receptor similarity matrix. So it's now saying, if I know which regions I'm connected to, and I also know how similar my receptor profiles are to other brain regions, can I predict function better? And we expect everything to go up because we're just adding a new variable into this regression. But the yellow points are the ones where if I add a spun version of this vector, so a randomized version in a way that preserves spatial autocorrelation, so that null model has now the same number of 2 input variables, right, instead of just one. If I add that null model, run that 1,000 times and compare against my empirical, I can see that my very specific, the specific topological distribution of this receptor similarity, it increases structure function coupling more than I would expect just by chance, just by adding a random vector to my model. Another thing I did, or sorry, here's my kind of summary thing. So here I see that this is kind of link. It's a pretty direct link between receptor densities and structure and function, and also that they mediate the relationship between the two, which was my main, like, I guess, hypothesis going into this project. I'm also just going to introduce this concept because it's going to come back later. So I had looked at functional connectivity, but I also was thinking, well, instead of just considering how two regions are functionally similar, what about just different kind of dynamical properties along the cortex? So brain maps instead of similarity matrices. And so these are MEG power distributions across different frequency bands. And each row here is a multiple linear regression model. I'm just taking the receptor densities. I'm trying to predict my sort of power distribution map on the cortex, trying to understand if the places where different receptors are has an influence on the types of oscillatory activity that we see across the cortex. And we see that, I mean, first of all, these R-squares are just like incredibly large. Also, if I just do the same model, but rotate all my input variables. And then here's a dominance analysis, which essentially what I do is I take the R-squared of this model, and then I split it up into each of the input variables to get a sense of how much is each input variable actually contributing to my model. So the way to interpret this is the sum of these dominances or percent contributions is equal to the total R-squared over here. And so it kind of distributes the contribution of every variable. I'm not going to go into that too much, but I wanted to introduce the analysis because it's going to come back later. Another sort of side, this was like I had worked with gene expression data before, and so a very natural question was, well, I'm doing all this receptor work. Would I just see the same thing if I used gene expression proxies? And this is something that was sort of popular in the field. We didn't have receptor density, so we would use gene expression as a proxy for the receptor density, which makes a lot of sense. I mean, you could sort of imagine that like the next best thing would be the gene transcript levels. And I just did a very simple correlation between the different receptors. So this is coming from PET imaging, and then from the Allen Human Brain Atlas, these are microarray derived expression levels of different genes, the gene that codes for this receptor. I'm just correlating them. So this is sort of very simple. And everything that's blue, there's not a correlation. And the ones that are yellow, there is. And what was sort of, I don't know if surprising is the right word, but I mean, first of all, most of them are not correlated. So it was not the case that we could just assume that a gene would be a good proxy for the protein. And there's many reasons for this. I mean, some of them methodological. It could be a problem with the data. It could be something about individual variability, all this. There's also plenty of biological reasons, things like protein turnover, different regulatory mechanisms. And then the ones that are correlated, they're just like so nicely correlated that it's really hard not to believe that this is a real thing. So those are sort of nice to see. And then here's this analysis where I'm saying, well, how big is the correspondence between gene expression and receptor density? And then what is the differential stability, or essentially the stability of the gene across the donor brains? So how much is it varying across people? And there's this really nice relationship of, if I have a gene that's going to look different in all my different people, it's not going to be very correlated with some group average receptor density. That kind of makes a lot of sense. Whereas if you have a gene like 5-HT1A, for example, it's the serotonin receptors, this one, no matter where you look at it, the gene expression profile looks the same in all these people. The receptor density looks the same. Doesn't matter what tracer you use. Doesn't matter if it's autoradiography or PET. It just looks the same in everybody. And so it seems to be this really ubiquitous marker that's very stable across people. And, I think, of course, there's a lot of important work to be done in individual variability and how people are different from one another, but there are many ways in which we are the same also. So we would expect that there are some features that are quite consistent across people. So this is just sort of a note of caution. If you can use the direct receptor density, if you're doing dynamical modeling, if you're doing any sort of other thing where you want to get this measure of what is the presence of this protein that's a signaling receptor, I would definitely recommend using as direct a measure as you can instead of the gene expression. And then I think this is more to do with individual variability than data quality, because of all these sort of other supplementary analyses that I did. So at this point, that project, I was thinking about, these receptor densities. To me, they were a sort of very obvious molecular feature, very local molecular feature that would be mediating this relationship between structure and function. And then around the same time, I learned that synapses are diverse and differentially expressed in the brain. So this is very new work that is hopefully going to be preprinted soonish. I had started thinking about this at the same time I was doing the receptor work, so that's a few years ago now, but I didn't actually start doing the work until a bit more recently. That's why there was a bit of a time delay. But essentially, there's this work from a group in Edinburgh, Seth Grant, and he does a lot of synapse type mapping. And what that is that he's looking at, he's discovered that there are a bunch of different types of synapses in the brain. So to me, prior to learning this, I thought excitatory, inhibitory, and then broadly modulatory. That was my kind of basic understanding of different types of synapses. And it turns out there's all these different, even within different excitatory synapses are going to have different morphology. They're going to be expressing different proteins. In the same way, I was thinking, if you have a certain fingerprint of receptors, you're going to be responding to signals in a different way. Same thing with synapses. If you have a synapse that's sort of built in a way that it can better respond to one signal than another, that's going to be a synapse type. That's going to be a synapse that is functionally doing something different than its neighboring synapse. And so they're doing this in mouse brains. So they're looking at mice that are fluorescently expressing specific proteins. And so just to give you a sense of what this data looks like when I work with it, what I have is here on the rows, these are different synapse types. So you can see they've kind of been bunched up into these two groups. PSD95, that's a protein called postsynaptic density 95, is one of the markers that they are tagging to identify a very specific type of synapse that expresses this very specific protein. And another one is SAP102, that's synapse-associated protein 102. Both of these are postsynaptically expressed. They're both expressed at excitatory synapses, and they both have sort of a lot of mouse literature and a little bit of human. It's starting now, like also with the human work, but mostly mouse literature about the function of these synapses at the microscale. So things like they're going to have different dynamic signatures. They're going to have different effects if you knock out that gene, and then the mouse, how does it respond behaviorally? How well does it learn? How is plasticity affected? These sorts of things. But of course, this is all either at the microscale of a single cell, or it's at the scale of the behavior of the mouse. So I was sort of interested, well, in the similar vein with the receptor work, what happens if we start to map these different synapse types across the brain? How are they related to brain structure and function? So here we have these different synapse types. What they've done is they have this one protein marker. So these are PSD95 synapses. And then within PSD95 synapses, they can cluster them according to different morphological properties, so things like size and shape and whatnot. And that's why we have more than just one row representing PSD95, and same with SAP102. Now, the thing is, I didn't really want to work with, 11 different subtypes of PSD95, of which I had no idea their functional relevance. All I knew was that they had been clustered according to kind of morphological properties. So what I did is I just did a correlation matrix of how similar are these different subtypes expressed across the brain so that I can justify averaging. And so for SAP102, we see that they're all expressed. You can even see it here. They're all expressed quite similarly. So I just take an average. I'm working with SAP102 synapses. That is one synapse type. It's going to do a functionally specific thing. I'm just kind of treating that as its own thing. And then for PSD95, I really wanted to just average it all and have one PSD95 synapse type. And then I actually did do that. And then a co-author said, well, but look at this. You can really see that there are two different, you can even see it in this matrix, right? You can see that there are two patterns. Like, what are you doing? Why are you averaging across everything? So I don't know about you, but sometimes I need somebody else to tell me, like, I know what the right thing is, and I don't necessarily want to do it until someone is like, well, of course you need to do that. And then I'm like, yes, you're right. I need to do that. So I see that there's these two clusters. So now I have these two different, I have this one, cluster of PSD95 synapses. They follow this specific expression pattern. And this other cluster of PSD95 synapses, they follow this other expression pattern. And I didn't know their functional relevance. I mean, which is why I really wasn't happy with this sort of dichotomy, because to me, they're just PSD95 synapses. But then I dug a little further. And so this paper, this is a 2018 paper where they're first mapping synapse sites. Later in 2022, they do this really cool experiment where they're looking at the lifetime of a synapse. So this is another thing I had no idea was a thing, that synapses have lifetimes. But synapses aren't just, I mean, it makes sense if you think like synaptic plasticity, that synapses can strengthen and weaken and also just completely be recycled. And there's some turnover for these proteins and for these structures. And so what they were doing is they were mapping for the PSD95 synapses. They're seeing how long is that synapse hanging out before it gets eaten up. And what I find is that these two clusters that I had, these were just completely data-driven, like derived in a data-driven way. I find that they nearly perfectly align with the lifetime. So here, this bar plot is lifetime of synapse. So we have that this cluster, all those synapses that are expressed this way across the brain, all of these have longer lifetime. So they hang out in the cell for longer. You might expect them to be more related to something like memory. And then these shorter turnover proteins are expressed this other way in the brain. That's this cluster over here. These are ones that you might expect to be related to something like cognitive flexibility or something that requires you to really forget what you have learned on a quicker time scale. So that sort of gives you the maps, just so like we can be on the same page in terms of data. Here are the maps that I'm looking at. So I have long lifetime PSD95 synapse density. So again, PSD95, that's just a protein, a synaptic protein. It's marking a specific type of synapse. So we would expect that the synapse does its own thing. It has long lifetime. Same thing, PSD95 synapse density, but short lifetime. And then the third one is this SAP102 synapse density, where they did not do any measurements of lifetime. So I honestly just don't know how the lifetime of SAP102 synapses compares. This plot up here is just telling you which brain regions I'm looking at. And so I'm plotting the same thing twice. So this is a mouse brain, axial, coronal, sagittal views, and then duplicated. But these two parcellations are slightly different. One corresponds to the structural data, so track tracing data that I'm going to compare it to, and the other one corresponds to fMRI data in awake mice that I'm going to be comparing it to. So that's the data. Hopefully that makes sense. And the kind of key question I wanted to ask here, they've done experiments at the level of the individual synapse, doing things like patch clamp experiments and looking at the electrophysiological responses of specific synapse types. And so I wanted to understand if we have this brain map, this mouse brain map of different synapses and the density of the different synapses across the brain, what do the dynamics look in one brain region versus another brain region that has high versus low synapse density? So here's some time series. We're starting from low synapse density to high synapse density. You can see, I don't know, I've stared at so many of these time series that I don't know to what degree other people can see different patterns in them. But at least you can see that they're qualitatively different. And I think it's much more clear for SAP102, where I have this brain map of synapse density. I'm just picking some regions that have high versus low. So we go from low to high. You can see as we go from low to high, there is something, the time series, not only are they qualitatively different, there seem to be some features that are aligned with having more of the synapse or less of the synapse. And the idea here is if you have like a type of synapse and its morphology is such that, or the receptors that it expresses or whatnot is such that it's easier to respond to a certain type of signal, but it's also easier for it to create a certain type of signal. You might expect this relationship between where the synapses are and the types of dynamics that are in these regions. So to do this a little bit more quantitatively, we used HGTSA. So this, you might be familiar, Ben Felcher made this like toolbox. I think it was the bulk of his PhD work was to like kind of put this whole thing together. And it calculates about 7,000 different time series features on time series from different brain regions. So here we have all these different brain regions from the mouse, something like 7,000 time series features. This has been averaged across mice. And it's because I didn't want to just stare at the time series and say, what do I think is happening to these time series? I have something a little bit more quantitative here. I'm just doing a simple correlation between the time series features and these density maps. So I say, is there a relationship between the density of these different synapses, so where they are in the brain, and then what types of time series features we are looking at, where we seem to observe? And the PSD95 synapses, so these ones over here, are correlated with features of stationarity and model forecasting. So this is like the predictability of signal, such that if we have high density of these synapses, the time series are more erratic. And if you have lower density, they're more regular. They're more kind of cyclical, more periodic. And for SAP102, this one is so much more obvious to look at. I should maybe even lead with this. But here, it's very much correlated to these outlier events. So if you have low density, this is what our time series look like. They're more kind of stable. And then if you have these higher density regions, you suddenly get these peaks in the data. I should maybe mention that this data fMRI data set is coming from Alessandro Gozzi's group. So it's a fMRI data in awake mice, which I guess, I mean, also you'll see later in this project, but I think much of the fMRI literature in mice is in anesthetized mice. I didn't work with mice before this project or synapses, honestly. This is very, very typical of me to kind of, do one thing and then do something completely unrelated next. But in any case, you can sort of see the theme of the types of questions that I'm asking. So here again, we have a structural connectivity matrix. This is track tracing, so it's directed. And I'm just asking, well, if I have a specific type of synapse, and now that I've seen that, well, these different synapses have different dynamic properties. So if they're going to have different dynamics, is that going to manifest as broader macro scale patterns in connectivity in the way that they're structurally connected to each other or the way that they're functionally connected to each other? So here's just a very simple correlation between the synapse density of these different synapse types and in this case, this is out degree. And the reason I'm only showing out degree, I can sort of say this quickly, but in degree, because these are postsynaptic markers, you would expect that higher density of these synapses just means greater in degree in general, right? Because it's postsynaptic, so that means that there is a connection that's going into that cell. And that is the case. I find that across the 3 different synapse types that I was looking at, so the long lifetime, short lifetime, and then SAP102, they're all correlated with in degree, which was sort of nice because they're not very correlated with each other. So sort of was cool to see that we have this one, axis that overlaps across all these different synapse types. But for out degree, that one is a little bit less direct. So there, it's like it means if you have a synapse there, it's a postsynaptic protein, so I'm more likely to respond to certain types of signal that also make it more likely for me to connect to another neuron. So that's what that would mean. And here I find that for the long lifetime PSD95 synapses, there's no correlation at all. And then for the SAP102 synapses, there's a significant, weak but significant correlation. So I thought, okay, well, I have this dichotomy between my PSD95 synapses and my SAP102 synapses in terms of both the dynamics that they present, but also their connectivity profiles. And this is also replicated if I look only in the isocortex where I have data across different layers. I actually find that the out degree for SAP102 is correlated with synapse density in the layer, in the output layer of the cortex, so layer 2/3 for cortical connectivity, and the in degree of structural of having different connections is correlated to synapse density in the input layer, so layer 4. But only for SAP102, not for PSD95. So it doesn't matter what PSD95 is doing. It seems to not really have any relationship with connectivity, with structural connectivity. I should maybe mention at this point, here I'm only showing long lifetime and SAP102. I've sort of ignored short lifetime. And you'll see a theme throughout is that the short lifetime synapses, I just was not getting, it was very frustrating. I was not getting anything with them. Like they were just not behaving. And I'll talk a little bit more about that in a second, but that's why we're missing one of them. So here we have functional connectivity, and very similarly, I'm just correlating synapse density with degree. And here it's kind of have an opposite result where for the structural connectivity, I was getting no result for PSD95 and I was getting a result for SAP102. And for functional connectivity, I get a much stronger result for PSD95 and a weaker result for SAP102. So I sort of thought, is it possible that I have one synapse that's more related to forming actual physical connections and another synapse that's more related to the dynamics of these different brain regions? So at this point, this is where I talk about the short lifetime synapses a little bit. I was very frustrated. I was like, okay, I have this long lifetime PSC95 synapse. It's related to the predictability of a signal. If it had greater density, it's more noisy. It tends to be related to brain regions that have greater functional connectivity. If I, for these SAP102 synapses, if they're present, I get these big outlier events. Their presence also indicates more structural connectivity, both in and out degree. And so I have this kind of dichotomy function versus structure for these two different synapse types, and then short lifetime synapses behaving. And I thought, is it possible that it's because these synapses, because they're being recycled on a much shorter time scale, they just aren't good at establishing long-term connectivity patterns that I'm measuring with something like track tracing or fMRI. And so I sort of thought, well, I would imagine, like, I don't know, if I were to design a brain and have some short lifetime synapses, those synapses are probably the ones that are more related to cognitive flexibility or these kind of functions that sort of need to be quick on your feet. And so to try to get at this question, here we have the same structure function coupling analysis I did for the receptor work. So I've already showed this where I'm saying I have a structure function coupling. If I'm just using structural connectivity and just functional connectivity, then I add in receptor information and see how it improves. Same thing here, except instead of adding in receptor information, I'm adding in the synapse density information. So I'm saying, given my connectivity profile, my structural connectivity profile, can I figure out what my functional connectivity profile is if I also include information about what synapses are there? That's going to be relevant here. And I do this, here is functional connectivity from awake mice. So, okay, see that things go up, that's sort of expected, and sort of seems to go up for all of them. And I didn't really know what to make of this. And then I do it again for anesthetized mice. So this is a different data set of mice, but these ones are anesthetized. So they're not going to be, presumably, they're not going to be doing the functions that I sort of thought that the short lifetime synapses would be useful for. And then what I do here is I just compare the distribution for the three different synapse types. So short lifetime PSC95, short, that should say lifetime, not lifespan, short lifetime PSC95, long lifetime PSC95, and SAP102. And in the anesthetized group, the pink histogram is telling me how much does my R squared change when I added my receptor information. And the blue one is how much does my R squared change when I added receptor information for the awake mice. And what I want you to notice is that the distributions for long lifetime PSC95 and SAP102 synapses are very similar. They're kind of overlapping. And for a short lifetime, once we're anesthetized, that increase in R squared just shrinks. And so what I thought that this means is that the placement of these short lifetime PSC95 synapses, they're bridging this link between structure and function, but only if you're awake. So if you're awake, they're useful. And if you're anesthetized, they're really not doing much for you at all. They're not going to be. Whereas the other two, long lifetime PSC95 and SP102 synapses, they're actually quite useful no matter whether you're awake or not. So I kind of think that these two synapses, especially the fact that they're related to these outlier events and also this predictability of the signal, they're sort of ubiquitous and they're important in regulating brain function regardless of your behavioral state. So that's my synapse work. So I mean, this is the only work I'm showing that's not published, which means that if you have feedback, I can actually integrate it. So, I'm happy to take criticisms. But that sort of brings me back to my intro here with this integrated brain. And when I showed you some work where I was linking receptor densities to structure and function, and then really similar work, honestly, in terms like conceptually very similar, very different in terms of data and in terms of it's mouse instead of human and all this kind of stuff, but conceptually very similar work with synapse densities. And then now, of course, the next thing I'm going to talk about is the brainstem, which really tries to show that a lot of these phenomena that I find in the cortex, because a lot of my work, not the synapse work because it's the mouse, so I was able to look at the whole brain, but a lot of my previous work was just cortex-centric, to kind of show that a lot of these phenomena that we see in the cortex can also be found in the brainstem. So this is maybe the project that you actually were here to see. I'll kind of walk you through it. The brainstem is my favorite brain region to ignore. It is like very easy to introduce. Everybody knows what it is. It takes like a big chunk of any intro to neuroanatomy class. It's, I don't know, everybody knows I don't have to convince you that it's important structure, but it's still like my favorite brain region to ignore or to represent just as a single node, like in any kind of structural connectivity matrix. I mean, I don't know for your data sets, but for my data sets, brainstem is often just like a single node. A lot of what we know about brainstem, given, it's kind of interesting that we know so much stuff about the brainstem, but then if you actually dig into what we know about the brainstem, most of that is coming from animal literature or histological experiments in postmortem brains. So we actually don't know very much about what is happening in the brainstem when in a living human, right? We have a sense of what it's doing in mice and in rats and lots of cell staining experiments. If you have a postmortem brain and you can like look through it and kind of lots of anatomy of the brainstem is very well established, but in terms of in vivo function, very little. And so really what's missing is this kind of whole brainstem approach to what is actually really going on in the brainstem. And so the main question that's driving this project is, I couldn't totally remove myself from being corticocentric, so it's still a little bit corticocentric in that I'm wondering how the brainstem reflects knowledge on cortical function. So there's of course a reason that brainstem functional imaging has lagged behind cortical functional imaging, and one of the maybe smaller reasons is that it requires an atlas or a template of brainstem nuclei. So a lot of people who are studying the brainstem, especially fMRI, brainstem work in fMRI, what they'll do is they'll have a very specific nucleus of interest, and then they'll delineate that nucleus of interest themselves. And so it's not necessarily standardized, it's not necessarily reproducible, and also it's just limited to the one nucleus that you delineated, because it's a lot of work to be able to delineate these well. Now, luckily, that has sort of been fixed. I'll tell you about that in a second. Another thing, of course, high spatial resolution and high sensitivity, because we're so deep down in the brain, and all these physiological noise reduction pipelines. The brainstem is really close to vasculature. It's very close to CSF. It wiggles every time your heart beats, which means that if you're imaging a living human, you're already going to get all these motion artifacts. So it's a big challenge to image. That's where Marta Biancardi comes in. So she is an expert in brainstem imaging, both structural and functional anatomy in general in the brainstem. She's a professor at Harvard. And for many years, she's been developing a probabilistic atlas of brainstem nuclei. And so what she's done is she's delineated. So for example, here we have the dorsal raphe and the periapical ductal gray, and she's been using T2 weighted images to get these probabilistic maps of where these nuclei are in MLI 152 space. So this makes it possible for anybody, like yourself included, to take data and then parcelate it to like 58 different brainstem nuclei instead of just treating the whole brainstem as one thing. And then a little later, recently, 2 years ago, she put out functional connectivity data in 20 people. This is data that's been specifically imaged and pre-processed in a brainstem optimizing way. So I think the only acquisition difference is that the head coil has been tilted back a little bit to improve SNR in the brainstem, but lots of pre-processing differences in terms of how we're dealing with noise and physiology and heart rate and whatnot. And so what this means is we finally have this atlas. So here are all my brainstem nuclei. There's 58 in total. This is how I'm going to plot it most of the time because it's just a little bit difficult to see what's what. So I'm just plotting the node centroids, and then the gray stuff is the cortex. You can just sort of see with respect to the cortex where the brainstem is. So we have 58 brainstem nuclei in total. It's 400 cortical regions that I'm looking at, although I replicated this in 100 cortical regions as well. And this is a very small data set of only 20 people with this brainstem specific acquisition and pre-processing protocol. The first thing I did was just to make sure that my brainstem atlas is acting the way I would expect. So first of all, each of these, that's a brainstem. So we have coronal, sagittal, and axial views. And then across all the different neurotransmitter receptor systems, so these are they're volumetric, right? So I actually have brainstem data for these receptor densities. I just parcellated them to the brainstem atlas to make sure that things are where they should be. So for example, for serotonin, that I'm getting that the raphe nuclei have the greatest serotonin density, or for dopamine, that the substantia nigra and the ventral tegmental area have the greatest density, things like that. So that seems to look good. And as a result, we have this functional connectivity matrix. So this is cortex, and then we have appended the brainstem. And you can see right away that functional connectivity is quite a bit lower when we have the brainstem involved, probably due to SNR. It's hard to get a correlation. Here we have cortical connectivity. It looks the way we would expect. This is ordered Schaefer 400, so you can see the resting state networks in there. And then here we have the brainstem data on top. And if you zoom into cortex to brainstem functional connectivity, this is of course a symmetric matrix, we do see these stripes. So there are these dominant patterns, both vertical and horizontal, right? So there are dominant patterns of connectivity between the brainstem and the cortex. That's what this means. So then what are these dominant patterns? So for every brainstem nucleus, here each point, each centroid here, that's a brainstem nucleus. For each one, I'm going to sum up its functional connectivity to the cortex. I'm just plotting it here. So darker colors and bigger circle means greater connectivity to the cortex. You can think of this as hubs, brainstem hubs of cortical connectivity. And what I like about this is that they're sort of the hubs are placed where you might expect them to be placed in the sense that they're the top, they're in the middle, they're the bottom. Kind of good design if you want efficient information transfer. One thing that maybe is not so expected is the locus coeruleus. I don't know how much you guys know or care about specific brainstem nuclei, but in any case, locus coeruleus, you might expect that to be a brainstem hub. I don't find it's a hub. It's quite small here. But yeah, so here we have this kind of where in the brainstem are we connected to the cortex? And then on the flip side, the question becomes where in the cortex are we most connected to the brainstem? So here for every cortical region, I'm summing up functional connectivity to the brainstem. And what this is telling me is that these anterior regions are much more functionally connected to the brainstem than these posterior regions. This connectivity profile, I mean, a couple things about it. One thing is that the regions in which they have more functional connectivity to the cortex kind of align nicely with different cell types, so site architecture. I actually think this is very related to structural connectivity. I don't have structural connectivity to the brainstem, so I haven't run any analyses myself. But just based on the literature, there's a lot of studies for track tracing in macaques. So of course not human, but in macaque brains, looking at these long projection fibers that go down to the brainstem and originate in these interior singular regions. So I do think that the reason we get higher functional connectivity between these regions and the brainstem is because they're supported by underlying structural connections. But also, this brain map reminded me a lot of those MEG power distribution maps that I had previously related to receptor densities. So in that receptor analysis, the question was, if we have different receptors, depending on the kind of spatial, the overlap of these different receptor densities, am I going to create a specific type of oscillation pattern that we can pick up with MEG? Here the question is, my input or output, it could be either, right? But you can think of this as a brainstem input map. Brain regions that have more connectivity to the brainstem, do they also show specific oscillatory dynamics? And you can sort of imagine if you have a lot of input from something like the brainstem, you might have different dynamics than a region that doesn't have a lot of input. And of course, with MEG, you can't really dig down into it's much more difficult to dig down into the brainstem. And so that was sort of nice to see this strong, especially for the alpha rhythm, which of course is the most dominant pattern, the strong relationship between brainstem input and this type of dynamics. Now, the main question I wanted to ask when I got this data was, do groups of brainstem nuclei make distinct connectivity patterns with the cortex? And so what I mean by that is if I take my functional connectivity matrix and I look at how is each brainstem nucleus connected to the cortex, do I find that some brainstem regions are connected in one way and other brainstem regions are connected in some other way? And can I sort of divide the brainstem up in this way? Now, I ran into a bit of a problem, which was that if I take, this node, so this would be a brainstem nucleus, how is it connected to the rest of the brainstem? Compared to another brainstem, sorry, this is a cortical region now, and how is it connected to the brainstem? There's some other cortical region, how is it connected to the brainstem? They all kind of look like that average or summed map, which is this dominant pattern of connectivity between cortex and brainstem. And another way of showing this is here. If I say for all of my regions in my atlas, so this is 400 cortical regions and 58 brainstem nuclei, or if we separated brainstem to cortex, how similar is that region's connectivity profile to the brainstem? How similar is it to my group average connectivity profile? And they're very high. These correlations are very high. So what that means is that all of my brainstem nuclei are kind of connected to the cortex, and all my cortical regions are connected to the brainstem like in the same way. So this was a bit disappointing because it meant that I can't find distinct patterns of brainstem nuclei that have different patterns of connectivity to the cortex. Now, whether this is biological, so whether there really is this really dominant pattern of connectivity, or whether this is kind of a noise issue, something like a sort of akin to a global signal, sort of this one dominant pattern. Is that a noise thing? Is it an imaging thing? Is it biological? I don't know. But then I thought, okay, well, this is not very satisfying. What happens if I just look at the residual? So if I regress that dominant pattern out of each connectivity profile and just say, how much is the brainstem connected to the cortex above and beyond that dominant pattern of connectivity, and likewise for the cortex? What do I see then? And of course, these residuals are relatively small because this dominant pattern is so dominant, these correlations are also high. But in any case, I've regressed this dominant pattern from my brainstem cortex functional connectivity matrix. Then I correlate my columns, brainstem nuclei, and what this gives me is a brainstem by brainstem. So these are brainstem nuclei on the rows and columns. It's a correlation matrix, and it's telling me how similar are two brainstem nuclei in terms of their connectivity to the cortex above and beyond the dominant pattern. So just to make sure that we all understand what's happening, if I had done this analysis, this correlation matrix without the regression step, it would just be very, very pink everywhere. It would be very high correlations. They would all be quite similarly connected to the cortex. But after the regression step, I see these clusters. And so I thought, well, okay, of course, what are these clusters? And how are they actually connected to the cortex? So here, what I've done is this is a again, I don't know how much you guys wanted the dirty details, but just in case you do, I'm using Louvain clustering. I'm going across a bunch of different resolution parameters. Here's one of the solutions that works well in terms of has a stable solution when I do consensus clustering across multiple repetitions. But there are other stable solutions as well. So I think this is the figure that's in the main text, but in the supplement, there's also the solutions when we get coarser clusters. So a 4-cluster solution, a 3-cluster solution, and here these lines are just telling you how these groups get merged in these different cluster solutions. So here we have these 5 different colors, and so each one is a cluster of brainstem nuclei. And again, so for example, all the green ones, they would all be connected to the cortex in a very similar way. All the yellow ones are connected to the cortex in a similar way and whatnot. But then, of course, well, how are they actually connected to the cortex? I'm not an expert in brainstem neuroanatomy, so when I look at this plot, I'm not thinking like, oh wow, all those yellow points must be doing this one thing. I have no idea. So I had to go into and look at the cortex. And so here, just to make sure it's clear, do I have a, yeah. So let's say if we take all the yellow regions, I'm just looking what is their summed functional connectivity to the cortex. That's the yellow plot right here. And then similarly for all the rest. So this would be the cortical profile of how these yellow nuclei are connected to the cortex, and then same for the green nuclei and the pink nuclei and the blue nuclei and whatnot. And what was really nice about these brain maps is that they're so recognizable. So I plotted them the first time I looked at them and I said, well, I already know what these are doing. I don't really need to dig deeper into the functional relevance of these. And I was very relieved that they were not like extremely noisy looking. I thought because of that regression step, I was quite, I didn't really think this analysis would work out. I kind of thought once I get these clusters, they're just going to be connected to like random, going to be very noisy looking maps. I'm going to have no way of kind of functionally identifying what these brain maps are doing. But instead, I get these really clear brain maps, and just so it doesn't have to be me, you trusting my eyeballs. I did a quantitative and neurosynth analysis. So here I'm just looking at what's the functional relevance of these brain maps? Where do I get functional activity for different tasks or different behavioral states or whatnot? And so here we see exactly as we would expect. This yellow one, we see some motor and visual regions, so we get functions like perception and multisensory engage, object recognition, things like that. The pink one is very limbic. We get lots of processes related to affect, related to emotion. And then we also have these like the blue, the green, the gray. They're a little bit more higher orders. We have some more cognitive related functions. We have some things related to memory. And a lot of these functions that in the history of, studying the brainstem, we generally don't think of the brainstem as being something that's involved in these more higher order cognitive processes. But of course, the brainstem and the cortex have so many connections between them. So naturally, one would expect the brainstem is supporting a lot of these functions. Then, of course, the question becomes, well, why are these brainstem nuclei connected to those functional networks? What is it about these brainstem nuclei? And this is finally where the receptors come in. So, of course, a lot of these nuclei are synthesizing the neurotransmitter, and then the receptors are expressed along the cortex. And so if you have this projection pattern that's sending out these neurotransmitters, and you have different receptors in the cortex that are there to receive that signal, we might expect that to be governing the relationship between the brainstem and the cortex. And so here's that dominance analysis. It comes back, and what I'm doing here is for each of these cortical profiles that represent brainstem connectivity of a different module, I'm taking my receptor distributions or my receptor densities in the cortex and predicting that map. And then I started to see all these kind of links, which I'll just sort of walk you through a few of them. But, here is the dopamine transporter. It is most dominantly contributing to the R squared of this green community's cortical pattern. And the green community is where the substantia nigra and the ventral tegmental area are. So these dopamine synthesizing regions related to this dopamine transporter. Same thing with something like norepinephrine. Here we have the norepinephrine transporter. It's most dominant in the blue community. Also, interestingly, it's quite dominant across communities, which I think does have something to do with this neurotransmitter being very related to like many of these processes. But in any case, it's most dominant in this blue community. The blue community is where the locus coeruleus is. So again, we have this kind of relationship. And this also happens for the serotonergic receptors and whatnot. And so I do think that there's this relationship between what these communities sort of get divided up by which neurotransmitter is being synthesized there. And then it gets connected to cortical regions that have those receptors that are there to listen to the neurotransmitter itself. And then the last figure in the paper is this kind of last almost for completeness kind of figure, which previously I'd looked at how are brainstem nuclei similarly connected to the cortex, and this is just asking the opposite. How are cortical regions similarly connected to the brainstem? And here we have this cortex similarity matrix. And because I'm using Schaefer 400, which is already ordered according to the resting state networks, the Yo, Creanin, like 7 resting state networks, I immediately saw this organization. And so I haven't done any clustering or anything on this. Like previously for the brainstem, do I even bother doing this? Yeah, here for this network here, that was me doing some clustering to find clusters in the brainstem. But for the cortex, I didn't do any clustering. I just did a correlation matrix, and I see these clusters very well. So I kind of knew if I take the first gradient of this matrix, I'm going to get a unimodal transmodal gradient. And this unimodal transmodal gradient, what's sort of fun about it is that we typically think of that gradient, unimodal transmodal gradient, which we get first axis of variation of cortical connectivity. So we think of that very much as how is the cortex connected to itself? How is cortical connectivity giving rise to these different unimodal versus transmodal processes? But this is not cortical connectivity. This is cortex to brainstem connectivity. And probably what it's reflecting is that the cortex really has this dominant axis of variation, and we see that projected onto the brainstem as well. And here, if I correlate the two, yes, they're very correlated. We kind of expect that. But not only that, the distribution of values is quite a bit more bimodal when we consider, I'm just going to move your faces, quite a bit more bimodal when we consider brainstem cortex connectivity rather than the cortex connectivity. And what I think that this means is that the brainstem is sort of anchoring these two poles. There's high connectivity to one pole, unimodal, and the other pole, transmodal. There's not that much happening in between. Whereas cortical connectivity is much more graded. It's much more smooth, much more continuous from these two different poles. And this we can also replicate. This is a gradient analysis, but I also did a community detection analysis with the Louvain algorithm again. And the funny thing there is that the only stable solution that I get is the one that divides them into two communities, these pink regions and then these blue regions. So I can only get a brainstem connectivity pattern to unimodal regions and a brainstem connectivity pattern to transmodal regions. I don't get stuff in between. And so then, of course, which brainstem regions are connected to these unimodal versus transmodal cortical regions, I get that this is unimodal connectivity is mostly to caudal brainstem plus the inferior colliculus. Inferior colliculus is one of the brainstem nuclei related to auditory functioning, so that sort of makes sense. It is a unimodal function. And then here, these rostral brainstem regions are most functionally connected to transmodal cortex, which is also sort of fun because these rostral regions, a lot of my neuromodulatory nuclei are there. Things like the locus coeruleus, the raphe nuclei, substantia nigra. And yeah, so those are all the main figures in my analysis. And then I get this question: what about the subcortex? You know, it's like, okay, Justine, like you've looked at the brainstem, and yeah, the brainstem's important, but obviously there's this like very important structure in between. And you know, I totally agree with you. That is very important. The problem is, so what I did was I repeated everything. Here's a supplementary figure. I repeated everything with the subcortical structures that I had in this data set. And you know, you can find things. You can say, oh, well, these subcortical regions are related to that and whatever. But I'm very hesitant to kind of dig too much into this result because the problem is the subcortical structures that I have, these free surfer regions, it's 14 subcortical structures. They're huge. They're cytoarchitectonically defined. It's very different than these brainstem nuclei that have been so specifically subdivided. And so when I say it's, you know, it's connected to the thalamus, like the whole thalamus, I don't even know how to interpret that. You know what I mean? Like, so I think this analysis would just work better. Well, first of all, if we do a better subdivision, but also once we have figured out how to kind of pre-process, you know, I think it makes sense that the cortex gets pre-processed one way, and then the brainstem gets pre-processed in one way. It's very specific to the brainstem, and each nucleus is very specifically delineated. And then the rest of the subcortical structures, I'm a little bit less sure. I'm like, I guess that would, be some sort of in between. I'm not sure, but in any case, I get the question, so I did the analysis, and if you're interested, you can dig into it. And then there's like, what about the cerebellum? And there, I'm so sorry, like I did not look at that at all. I have no idea. What about the cerebellum? This is Kaysen asking me, what about the cerebellum? I don't even know. Yeah. Anyway, so I have no good news there, unfortunately. But yeah, so to just summarize this brainstem work, the 17 fMRI brainstem data set lets us take something that I'm very familiar with studying, right? Cortical connectivity, functional connectivity, and then finally expand that also to the brainstem, which we know is important. And to get a sense of whether the connectivity patterns between brainstem and cortex, how are different brainstem nuclei connected to these different cortical networks, and realizing that these cortical networks are not purely cortical, they're also represented in the brainstem. I was looking at what might be mediating the relationship between the brainstem and the cortex. I think the neurotransmitter receptors make this very kind of obvious step, but it's not like I checked everything, so I'm sure there are other factors as well. And finally, that this unimodal transmodal cortical hierarchy is not purely cortical necessarily. I think it's probably anchored by brainstem input, probably either early in development or in evolution. But that's just me hypothesizing. And so, sort of big picture is that a lot of what we see in cortical function can be traced back to the brainstem. So that's my talk. How am I doing for time? Oh, a bit long, sorry. But I put everything on GitHub, like my code and data. So if you want to use any of these analyses or use any of the data, you can. And thank you very much for listening, and I'm happy to take questions.